So now I would like to invite Professor Chamindie Punyadeera for her presentation on democratizing healthcare access through salivary diagnosis. And Professor Chamindie is a pioneer researcher and advocate for women in STEM with a career spanning industry and academia. She holds a joint appointment in Science and Health at Griffith University and leads the Salivary Diagnosis and Liquid Biopsy Laboratory. She is also a principal research leader at the Institute for Biomechanics and Glycomics at Griffith University. Her work focuses on developing and commercializing biomarkers for heart failure and cancer. By validating biomarkers, her research aims to stratify patients by disease risk and treatment response, accelerating personalized healthcare solutions. Professor Chamindie, please.
Good afternoon. Thank you, Natalie. And is Whitey here in the audience? Thanks, Whitey, for the invitation. I think it's best to be inside rather than outside. It looks really, really miserable. And I have to say that I've been listening to these talks, the previous talks, and they're fascinating ones. And I must say that I'm looking very optimistic for my future when I age, because if I lose a limb or if I lose my, injure my shoulder, we have lots and lots of solutions for that. We went through a pandemic, and most of you must have done a nasal swab for COVID, right? Did you like doing that? No, right? Nasal swab was the first way to detect COVID antigen, and then came salivary diagnostics, and it was from Yale University spin-out, where they developed the first saliva test for COVID-19 antigen detection. It's pretty cool, right? Even I have done it myself. You spit into a tube, voila, in 2 minutes you get a result. To be honest, it was a struggle for me when I started this work about 14 years ago, trying to convince the Australian government we need alternative diagnostic testing to advance healthcare. It took me 7 years approaching a National Health and Medical Research Council to get research grant. Come COVID, I got 2 grants back to back, all for saliva testing. It just shows you that it takes a pandemic to realize that there are other alternative diagnostic testing and that type of research is really important. These are my disclosure statements, and I'm also the co-founder and director of Fragment Biotech, which we started last year, developing sensors for early detection of cancer and monitoring response to treatment in cancer patients, especially after immunotherapies. So the program highlights, I will go over the research program, and I will try and bridge cancer research into tissue engineering, into biomechanics. And we're very proud of 2 commercial products from my team. One is Cancer Detect, and it's the world's first saliva-based test to early detect head and neck cancers, which received FDA approval in 2021. The second one is Companion Diagnostics for heart failure, which is now licensed to an Australian startup company. And I will walk you through association between oral and systemic disease, how we can use saliva to detect cancer or heart diseases, and applications of saliva in heart failure, liver fibrosis leading to liver cirrhosis, and head and neck cancers. Australia spends about Australia spent $250 billion in healthcare in 2022 and 2023, and that's 10% of our GDP. And we heard today from Peter Chong, he said that 10% goes to orthopedic. Can we sustain a healthcare system like this? I don't think so. It's not, because every year 6 to 7% increases. So we need clever ways to bridge the gap in healthcare access and healthcare delivery. And hence, salivary diagnostics is going to play a major role in bridging that gap. we take biomarker from a discovery phase to validation phase, and we commercialize. And then we do bioassays. So saliva is a very viscoelastic medium, and it's not easy like blood to develop immunoassays to detect different biomarkers, but we have developed a workflow to do that. This is our NIH grant with Professor Chuck Henry, where we are developing a handheld point-of-care device to detect biomarkers in saliva to predict outcome in patients with heart failure. This is, I think you guys know, Associate Prof. R.J. Pandey from QUT, and we got this grant. Graham is in the audience. We got this grant from Department of Defense, and one of the aims is to develop a saliva denture to detect in real-time biomarkers for cognitive and physical performance of Australian soldiers. This part is the blood-based diagnostics or liquid biopsies, where we are harnessing the power of biomarkers present in blood and saliva. These are circulating tumor cells, circulating cell-free DNA or exosomes or extracellular vesicles as biomarkers to early detect, predict, risk stratify patients with cancer. This is how I see the bridge between cancer and tissue engineering or biomechanics, is that we are developing 3D model system to determine drug response in cancer patients. So before giving a drug to a cancer patient, we test to see whether the cells die. If the cells die, the patient would respond, because these recapitulate a patient's tumor tissue. These are tumor tissues taken and grown in a dish. Why saliva? I think to this audience, after a pandemic, is pretty obvious, right? And there are large number of companies now entering into the market. A month ago, I was interviewed by La Ponte, a French magazine, where they have, in France, they have developed a saliva-based test to early detect endometriosis. And there's also saliva-based tests to early detect prostate cancer. PSA is the current marker, which is not specific at all. And this COVID test you can buy from Walgreens for $40. Drugs of abuse, and this is the first FDA-approved test collection device that you spit into this collection device, and there is a buffer. When you close this, there is a buffer to protect DNA and RNA, and you don't need cold transport. This is the world first FDA-approved platform based on saliva to early detect HIV, and I believe you can buy it off Terry White pharmacies. This is the Australian invention from Newcastle, where they developed a saliva-based glucose test. And 23 and Ancestry.com is also using saliva. So for those early career researchers, it's very important when you go to conferences and stay around for networking events. You never know who you will meet, and you start conversation, and this is one of those conversations. 10 years ago, I met the CTO of Viome. Viome is like Microba. It's a health and wellness company based in US. It was a startup company where you send a poo sample and they will tell you what type of foods to eat. Then I tried to convince the CTO, and I said, have you thought about using saliva and linking it to oral and throat cancer? And then after a couple of months, we started working with the company where we collect a saliva sample, and we have developed a buffer to protect DNA and RNA in saliva. And these samples are then sent to New York, and they have a next-gen sequencing platform where we do gene expression, microbial gene expression. We know that the bugs in the mouth has an association for cancer development, especially for oral cancers. And then we heard a lot about AI and machine learning. So it's a bioinformatics company. They have used AI and machine learning to develop a diagnostic score based on 88 human genes and 182 microbial features. When you combine that, we have developed a threshold, and you can see the sensitivities and specificities of detecting cancer. And I think I can, so this, I call it kind of a, oops, how do I get this? Kind of a jewelry box. This is commercialized in the US for $399 USD, and it comes with clear instructions. Even my mom can, I gave this test to mom, even my mom could read it and understand what to do. So it's really important when you are commercializing things that it is easy for the general public to use. And it comes also with a FedEx. So once you collect the saliva sample, it's like a funnel. You just drool into the funnel, you tie the cap, and you send the sample. And within 2 weeks, you get results. So that's one of the products from my lab. And this is the second one where we developed companion diagnostics, and that is to predict outcomes in patients with heart failure. About 30% of patients after hospitalization, they get rehospitalized or they die of exacerbation or heart failure. How do we triage patients at hospital discharge to keep the patients who are at high risk and send those who are at low risk? Let's look at how you can, how you can use saliva to detect systemic diseases, because people must be wondering, how can you detect heart failure or heart diseases using saliva? Heart is somewhere else, salivary glands is somewhere else, right? Let's just look at a patient with a lung cancer. Lung cancer biomolecules get secreted. So let's say inflammatory biomarkers released from the tumor enter into blood. These biomarkers are then carried through the circulation to salivary glands via the carotid artery, and then they get released into saliva. So you can see the connection, how systemic events can be detected in the oral cavity. And if you look at the different mechanisms, how these biomolecules get transported, these are the salivary acini cells, and this is the endothelial or blood vessels. These molecules get diffused, like small molecules like cortisol, they can get diffused. Larger molecules, they undergo active transport, so it's an antibody receptor transport. And also some of the biomolecules get transported through the gingival crivical fluid. So we can literally say that any systemic events, biomarkers land up in saliva, and we can use saliva as a proxy for disease monitoring. This is a major problem, heart failure. Estimated 64 million people diagnosed with heart failure. A person 40 years of age has 1 in 5 lifetime risk of developing heart failure. 1 in 4 heart failure patients aged 65 years are re-hospitalized within a month and costing a lot of money. This work actually started when I was at UQ, where we use saliva to early detect heart failure. So what we did is that we collected saliva, we isolate protein, and we did mass spectrometry. And then we had a validation cohort, and then we identified these 6 different markers. If you look at here, this is the control group with heart failure class 1, group with heart failure class 3, 4. You can see that you can discriminate. And then we develop a model system to predict early heart failure, and you can see that these are the sensitivities. So area under the curve of 1 means that specificity and sensitivity is 100%, which you never get in biological validation studies. And this is work ongoing. This is our NHMRC grant, where we can use salivary galactin-3, which is a fibrosis marker, to correlate survival in heart failure patients. Let us look at this curve. These are the Kaplan-Meier curves.